In this video, we will discuss about de-immunization. So, what is this de-immunization and what is its purpose? Let us first understand that thing. Whenever we take any biologically developed medicine, there is a possibility that that medicine might generate immunogenic response because of the non-self epitope that are present on the biologically developed medicine. As we know that non-self epitope are considered by immune system of a individual as harmful, so they will develop immunogenic response and try to neutralize or kill those non uh, protein containing non-self antibodies. So it is essential that this immunogenic response should be minimized, or ideally the no immunogenic response should be there whenever a person take a medicine that is biologically developed, or any medicines, and whenever a person take. So there are strategies that are being formulated to make or to reduce this immunogenic response. So let us first understand uh, how this immunogenic response are developed. Although we have understood this in our previous videos, let us quickly go through it so that the people there who have not watched our previous videos are also on the same page. So these are the biologically developed medicine that are taken by the individual, or that are uh, that is presently inside the individual. And uh, when this biologically developed medicine come in contact with CD4 T cell, because of the non-self epitope, it will generate uh, sorry, we have to go through, we have to start from here. This is the latest step. So this is the biologically developed medicines. So when they are at this moment, they have non-self epitope. So it is first detected by the dendritic cells, and dendritic cells, since it detect the non-self antibodies, it will try to engulf this biologically developed proteins or medicines. And in this process, this antigen presenting cell will convert into the dendritic cell will convert into the antigen presenting cell, and it will uh, provide this non-self epitope to the CD4 T cells, and these CD4 T cells will then release cytokines. And uh, when this antigen presenting cells provide these epitopes to the CD4 T cells, the CD4 T cells get activated after receiving two signals. One is through major histocompatibility complex, and second is the co-receptor signals. This active after that the CD4 T cells get active, and this active CD4 T cells will release cytokines, and that will generate more CD4 T cells, or we can say adaptive immune response against this non-self epitope. At the same time, it will also increase the B cells, and these both B cells and T cells will act against these biologically developed medicines. And as this because at this moment the immune system of the body will produce ADA, that is anti-drug antibodies, the antibodies that can target the drug that has been injected by the that has been taken by the individual. So our final objective is to reduce this formation of anti-drug antibodies. So what approach people have taken is that they do a whole experiment in vitro analysis. So what they will do is original biotherapeutic drug candidate, that is the medicine that is to be taken by individual, will be uh, will come in will we can say will inject will do an in vitro injection in the lab and try to identify which part of this biotherapeutic drug will interact with major histocompatibility complex. We will use major we will use patients blood samples and this original biotherapeutic drug candidate, and we will see uh, which part of this biotherapeutic drug candidate will interact with, or which peptide of this biotherapeutic drug candidate interact with major histocompatibility complex 2 of the patients. And from that from this step 1 we will get the information about the peptide that interact with the biotherapeutic drug candidate. And after that we will do a alanine scanning to identify uh, which amino acid among the identified peptide will interact with the biotherapeutic drug candidate. And then we will use or the process will use different we try to substitute different amino acid against those uh, amino acid against the amino acid identified in step 2, and try to see the interaction of major histocompatibility complex 2, at the same time the function of this biotherapeutic drug, and the combination at which both of them uh, fulfill its requirement, that is there is less reduction in the biotherapeutic drug application or function, and there is more increase in the non-functional of this peptide against this major histocompatibility complex. That combination will then be used will then by individual, so that it will develop less anti-drug antibodies. At the same time, it uh, the purpose of this biotherapeutic drug will also be fulfilled. This is how process has been this project or this deimmunization process uh, is planned. However, this study is still under process, and it is yet to be approved by FDA for normal use. I hope you have understood what this deimmunization means and how it will improve the function of the function of these biotherapeutic drugs, and as it will reduce the side effects that are being generated. That's all for this video. Thank you for your time.